Hey, my name is Luke Perry. Today is 10/27/2020. Hope you can see that. So, my presentation today is on fat absorption and its enzymology. So, when we digest fats, we're ingesting hydrophobic materials that aggregate with each other and don't dissolve in aqueous solutions, like our stomach. The chyme that enters the small intestine looks like salad dressing, with a bunch of fats mixed into the liquid. So it would have that layer. There's layers of fat that you can tell that there's a difference in fat and liquid. That's what our chyme would look like. And so bile is actually the thing that we use to break those things down in the human body. Um, so bile is made up of three things: bile salts, bile pigments, and cholesterol. So bile salts are the main ingredient in what makes fat soluble in aqueous solution. They are made up of steroid bile acids and amino acids. Bile pigments are made up of one compound, mainly bilirubin, which comes from the breakdown of red blood cells. Um, so that's another thing. The breakdown of red blood cells goes into the bile. Cholesterol is just cholesterol and is excreted into the feces. So here we have a really good diagram of what it looks like to create bile and where it's stored and its excretion. So right here we have the liver, and underneath the liver, on the posterior side of the liver, the gallbladder is there, right right here. That's the gallbladder. So the liver is actually creating the bile and it's sending the bile into the gallbladder through the common hepatic duct right here. And it's stored here in the gallbladder. And then when fat enters the duodenum, which is right here, it triggers the contraction of the gallbladder here. And to release the bile through the common bile duct down into through the sphincter of Oddi, um, and releases that bile onto the fats so they can be absorbed. So, bile salts are amphipathic. They have two sides to them. They're hydrophilic and they're hydrophobic. So right here is this hydrophilic side that associates with water, and that then there's the other side of that, which is the hydrophobic side, which associates with lipids. And so that double-edged sword is able to combine together and coat these lipid droplets and make smaller emulsions in the liquid. So it's breaking it down even further from those larger emulsions. It's really important that the bile salts have that hydrophilic region because it allows fats to dissolve in water more readily. Um, so here's the thing: is once those um, bile salts are surrounding the fat, they need to be broken down even further, and these major enzymes are involved called lipase and colipase. So colipase is secreted as procolipase in the pancreas, which is once it enters the duodenum, procolipase is converted to colipase by trypsin. Um, and this colipase actually breaks down the bile salts a little bit and displaces them so that the lipase can access the soluble fat emulsion. The reason that the lipase needs access is because it breaks down triglycerides inside the fat emulsion and breaks them into monoglycerides and two free fatty acids. We'll see a little bit more what that's like in the next few slides. So there's other enzymes as well called phospholipase, which breaks down phospholipids into more singular entities. cholesterol is absorbed intact. There's no enzymes that break it down. So here's what happens once the enzymes get a hold of the fat emulsions. Micelles form. Micelles are small discs that are filled with fats. So on the exterior of these micelles, you have bile salts right here, bile salts, phospholipids, diglycerides are right here. You have monoglycerides, and you have within the actual micelle itself, you have cholesterol and free fatty acids. These cholesterol doesn't have doesn't have a hydrophilic region, so that's why they actually congregate on the internal side of the micelle. Now here is the whole kit and caboodle. So from previously, we have the bile salts that are coming from the gallbladder that that form emulsions from the large droplets of from the stomach, and then from those emulsions you have the enzymes that come and break those emulsions down into micelles. From those micelles, what's really important is the two main things that come out of it are monoglycerides, and free fatty acids that diffuse freely into the cells of the small intestine. The cells of the small intestine are also called enterocytes, which is a good term to know. So from the enterocytes also, the cholesterol is actively transported into those enterocytes. And the bile salts are actually reabsorbed in the enterohepatic system. So at around the distal ileum is when before the large intestine, the bile salts are reabsorbed and brought back to the kidney, or brought back to the liver. So these fats, once they enter into the enterocyte, they go directly to the smooth endoplasmic reticulum, whereby they form chylomicrons with cholesterol, triglycerides, and proteins. And these chylomicrons are huge molecules, huge balls of fat and protein that must go to the Golgi apparatus and get a vesicle to form around it so that it can leave the cell via exocytosis. And the thing is these chylomicrons are so large that they can't enter into the capillary system. They must go through the lymphatic system because the lymphatic system has those larger openings. And one of the things called a lacteal, which is the lymphatic vessel of the microvilli of the intestine, is there to accept the chylomicrons. So the chylomicrons then go through the lymphatic system and into they exit near the right atrium of the heart, whereby they enter the venous system and they go throughout the rest of the cells and distribute their fats, the triglycerides mainly, into the cells for energy storage and other things. So that is the main presentation there. Also, what you have is smaller fats, really small fats that come in and diffuse into the enterocyte, can actually be diffused into the capillary, and they can go into the bloodstream directly. So that really makes up my presentation. I hope you enjoyed it, and I hope you learned a lot from it.